Yeah, good evening to all my dear students, and today we are going to start with one of the important unit which is called as the cell cycle and cell division. How the cell cycle and cell division, what are the importance of that, everything we will be discussing a brief about that one. Okay, this is a chapter which we are going to discuss. Under this we are going for the introduction part. What are the things which are present in the introductory part of this that we are going to discuss. Introduction. Here, see, we will see here, all the cells reproduce, all the cells which are present reproduce by dividing into two, with each parental cell giving rise to two daughter cells. Any kind of a cell division, we know that a single cell which is present will be a dividing and it will be producing a new cells. Okay, and that's why you find the single parental cell which is present here will divides and produces how many daughter cells? Two daughter cells will be produced. Like you can say this is a one of the cell and it divides and it produces the two daughter cells. The two daughter cells will be produced. This is the main essence of the cell division, how it will be dividing. Next one, growth and the reproduction are the characteristic features of a cells, indeed of all the living organisms. You find these are the two important characteristic features. What are the two important beta? Like growth and the reproduction are the characteristics of a cells. Generally the cell will be increasing in size, or it will be a developing, or the growth will be seen and it will be a reproduced. Reproduced means you find the single cell will be dividing and producing a two daughter cells. The two daughter cells are produced by the cell division. Generally in unicellular organisms we will be calling this one as what you know, it is called as division. It is a generally the division, normally a single parental cell will divide and produces the two daughter individuals. This is a division. But there are two different things. One, in multicellular organism we can consider this as a division, whereas in the unicellular organisms we can consider that one as a division, plus it is also considered as a type of reproduction. What kind of a reproduction? In the lower classes you have studied, like a single bacterial cell will divide by, or it undergo reproduction by binary fission. Like same kind of a thing, the single cell will be dividing and producing a two daughter cells. That's what we call here as the growth and the reproduction are the characteristics of a cells. Indeed all the living organisms is the same kind of a thing. It can be seen as a common for almost all the living organisms. Such a cycles of a growth and the divisions allow single cell to form a structure which consists of millions of cells. As we now think about one of the important thing, like every multicellular organism will start its life within a single cell which is called as a zygote. What is a zygote? A fusion of male and a female gametes by a sexual reproduction results in the formation of a zygote, which is a unicelled. This unicelled, the zygote will undergo repeated mitotic division. That is a very important. Once again I am repeating, repeated mitotic division which results in the formation of embryo. This embryo will undergo the differentiation and forms a multicellular organism which is having in a millions of cells. Is it right? This is the main aim of the division, like a cell division. What are the division? What are the things which are present? Where does it occurs? We have seen this one. The single cell divides and produce the two daughter cells. Generally the growth and the reproductions are the characteristic features, and also all the multicellular, it starts its life within a single cell which is called as a zygote, where the zygote will undergo repeated mitotic division and forms the number of cells. This is the introductory part. That is the main aim of the cell division. Now we come for the cell cycle. What is the cell cycle and how do the cell cycle will be performed, and what are the things which are present here? We are going to see this one. Like you come here, cell cycle. What is meant by the cell cycle? What are the things which are present in the cell cycle, and what are the importance of the cell cycle that we are going to watch it. See here the cell cycle, what are the characteristics which are given? What is meant by the cell cycle and how the cell cycle will be there? If you open an NCERT, you will be having a clear cut picture, or the diagram will be there for the cell cycle. And what are that? We are going to see certain kind of a points. It is a sequence of events by which cell duplicates its genome. Cell will duplicate its genome, synthesis the other constituents of the cell, and eventually divides into two daughter cells. We know if you take any kind of a cell, what are the things which are present in a cell? Cell is there, a nuclear part, the nuclear membrane is there, and you find the chromatin material will be present. There are certain kind of a cell organelles which are present inside the cell or suspended in the cytoplasm. The various kinds of things which are present here. What happens? Its genome gets duplicated, means doubled, and also you find the cell organelles which are present here will get doubled. The cytoplasm gets doubled, everything which is present here gets doubled. That means this cell will increase in its size. And the nuclear material of this, you find the two sets of this will be found inside of this. And here the cell organelles also, you find everything which is present in the cytoplasm gets doubled. That is what the first point says. Next one, we come for cell growth results in distributing the ratio between nucleus and cytoplasm. See here, I already said, here the cell which is present is having a certain kind of a number of chromosomes, certain amount of the DNA, and also the number of cell organelles which are present will be a certain number. What happens? All the things which are present there gets doubled. When it is a double, it can be easily distributed into the two daughter individuals. Is it right? It can be easily distributed into the two daughter individuals. Therefore, it therefore becomes essential for cell to divide to restore the nucleocytoplasmic ratio. That is also very, very important one. Say for example, nucleocytoplasmic ratio. Say cytoplasm which is present, it is called as of cytoplasm. What is the amount of cytoplasm? Suppose 100 ml of cytoplasm is present. Now after doubling, how much it becomes? You might guess it will be a 200 ml it becomes. Suppose here the amount of the DNA which is present is 20 picograms. Pico refers to 10 power minus 12 grams, which is called as of pico. 10 power minus 12 is pico. Picograms means 20 picograms. 20 picograms amount is present. What will be the amount here after the cell division here? Like when cell is increasing in its size, what happens here? This becomes 40 picograms. This becomes 40 picograms, doubled. Then what happens? From this parental cell, when the two daughter cells are produced, each daughter cell will be having in a nucleus, and the amount which is present here will be how many picograms? Can you guess? 20 picograms. Here also 20 picograms will be present, and the cytoplasm along with the cell organelles which are present, what will be the ml? The ml which is present here will be 100 ml, and here also 100 ml will be present. Did you understand? This is what called as for nucleocytoplasmic ratio must be equally distributed into the two daughter individuals, which is given here. Next one we come for next point, cell growth in terms of cytoplasmic increases is a continuous. Generally after that, the two daughter individuals are formed, the cell will start increasing in its size. Once again the daughter individual has to become a parent. Again that parent, there the amount everything which is present gets doubled, and again it starts dividing. Like this, one cell will divides and produces the two daughter individuals. Once again the two daughter individuals will once again become the parent, and again they divide, again they produce the two daughter cells. Like this it will be a dividing. The process and the duration of cell cycle can be vary from organism to organism. Like for the cell to become two daughter individuals, the time taken by them will be varying from one organism to another organism. It is not constant for all the kind of cells, because in the nature we have two kinds of cells, which is called as a prokaryotic cell and eukaryotic cell. The prokaryotic cell which is present requires the less amount of the time for dividing, Whereas a eukaryotic cell, you find the majority of the membrane bound cell organelles are there, and also it takes much time. For example, one of the example is given for us, like we are going to see for the unicelled and a eukaryote and a prokaryotic kind of a thing. Like you can say, is the cell which is present here, the cell cycle, the duration is how many? How much time is this? This is called as of 90 minutes it takes. And the next one, human cell cycle duration which is present is how many hours? 24 hours. Like in us, for the dividing of a cell, one cell will divide and produces 2 daughter cells in a humans, and the time taking for this one will be at 24 hours. In this 24 hours, like 23 hours is the one which will be used by the interphase. Rest of the 1 hour is used for the division purpose. That's what which is given here. And another point, can you check it? The cell division proper lasts for only about an hour. That means only 1 hour is required here. Like this, you find the cell will be a dividing, and this is called as the complete essence of the cell cycle that we'll see in the diagrammatic representation in the next page. The next page. Here, any kind of a cell cycle which is performing, or the cell cycle, the phases which are present here, you find the cell cycle is having 2 important phases. One is called as interphase, another one is called as M-phase. These are the 2 important things. Just now I said, like in humans, it takes how many hours? 24 hours. Out of this 24 hours, 23 hours I said like it will be used by one of the stage which is called as interphase, means it will be taking a 95% of the total duration of a time interval which is required for the cell division. How much time? 95% of the total duration of a time interval which is required for the cell division. Which one will be taking, and how much it will be consuming? 95%. And here the M phase which is present here will be lasting for the 5% of the total duration. Totally now it has become 100%. Like this, you find it will be taking place. The first stage which is present is called as of interphase. What is meant by this interphase? What are the things which will be happening in the interphase? What is meant by the M phase? What are the things which will be happening? We are going to see. Called the resting phase. It is called as a resting. Why it is called as a resting? Because it's a non-dividing phase. Non-dividing phase which is present. That's why it is called as a rest. Rest means you find it, the cell along with the cytoplasm, everything which is present, it is not getting divided. It is a resting phase, but it is considered as metabolically active phase. So you can see cell is preparing for the division by undergoing a both cell growth and the DNA replication in an orderly manner. What happens? What happens here? You find the cell is preparing for the division by undergoing the cell growth will be happen, and also the DNA replication will be done. DNA replication. What is meant by this DNA replication also? I will be giving. One double standard DNA which is present there will becomes a 2 double standard DNA, because it has to distribute the nuclear material into 2 daughter individuals alike or the same. That's why it undergo replication. DNA replication is an orderly manner. How it happens? That will be a watching here. It lasts more than how much percentage? Like I already said, how much? 95% of the total duration of a time interval which is required here is used here for the cell division of the duration of a cell cycle. Now we come for the next part called as M phase. What is meant by M phase? Actual cell division phase is this. Actually, what is this? Cell division phase. What is this? Starts with nuclear division, which is called as of karyokinases, and usually ends with cytoplasmic division. The 2 points are the one which you have to note. One is called as first one which is present is called as, what is that? First one is called as, it is called as karyokinases, and the next one is called as cytokinesis. What is meant by the carry-on? Carry-on refers to what? Nucleus. Kinases refers to division. Means here it is called as of nuclear division. Nuclear division. This is called as a nuclear division, and this is called as cytoplasm division. Plasm division. This is the actual a dividing phase which is called as M phase. M phase is actually a dividing phase where the cell divides. And how much time it requires? Just 5 minutes. It requires just 5%, not the 5 minutes, 5%, sorry, 5%. Even though it is a 5%, many events which are present there are we are going to watch. Many events are going to happen. Much time for the explanation is required here. But for the interphase, even though it is a 95% of the total duration, but we will be studying that one in only the 3 phases. The interphase which is present is mainly divided into 3 important phases which is called as G1, S and G2 phase. G1, S and G2 phase. What is this G1 phase? After this S phase. After this G2 phase. What are this? This is called as G1 or growth 1 phase. Growth 1 or gap 1 phase. S stands for synthesis phase. Synthesis phase. And G2 is called as, now you can guess what is this? Growth 2 phase. Growth 2, or it can be called as gap 2 phase. Gap 2 phase. These are the 3 stages which are present. In the next one, the diagrammatic representation of that, I already said that we are going to watch it like a interphase and M phase. And here the M phase, the karyokinesis is one of the important thing which is called as a nuclear division, and the cytokinesis is called as a cytoplasm division. Nuclear karyokinesis is nuclear division, which is further divided into 4 stages, of which the first one is called as, you find the first one called as, what are the first one of this which is called as prophase, next one is metaphase, next one is anaphase, next one is telophase. These are all the phases or the stages with the diagrammatic representation. In the next page I am going to show to you. Please watch it. This is a complete part which is present. Here I told the interphase. You remember this interphase in the last page we have seen this interphase. What is this interphase? It will be a lasting for how much percentage? 95% of a total duration of a time interval which is required. I have already given here the divided further into 3 phases which is called as an interphase. First one is called as a gap 1 phase, next one is a S phase or synthesis phase, next one is a G2 or the gap 2 phase, which can also call as a growth phase is called. What do you call? Growth phase can be called. This is the complete or the entire diagrammatic representation which will be present there. You can say, you can check it here. This is the starting part where the cell will be entering. This is the starting part, start, where the cell will be entering here. And it will be completely coming and it covers the interphase. This is the complete part which is present here is up to this part, up to this, which is considered as what phase? Up to this is considered as what phase? Interphase. See here, this is a complete interphase. And how many are there? G1, S, G2 phase. G1, S and G2 phase are the 3 things which are present here. Chalo, we'll come here. After this, the next phase you can check it here which is given in the diagrammatic representation which is called as a M phase is given. You can check it here. What is this? M phase is given. This M phase is mainly divided into 2. In the last page we have seen. What is that? M phase is divided into 2. One is called as karyokinesis. Another one is called as cytokinesis. Karyokinesis is further divided into 4 stages which are present, I already said, but here it is mentioned in the diagrammatic representation. First one is called as a prophase, next one is a metaphase, next one is anaphase, next one is a telophase. These all 4 stages will be coming here for this karyokinesis part. That is a one part of M phase. After that, the next stage which is present here is called as a cytoplasm division which is called as a cytokinesis. After the cytokinesis, single cell which is started, it comes all the way through this cycle and finally it will be coming as a two daughter individuals are produced at the end. You can check the same kind of a diagrammatic representation is given for you in the NCERT. The same kind of a diagrammatic representation is given for you in the NCERT. This is very important one which is given for you in the NCERT. You can check it very clearly. This is the same kind of a part which is given for you in the NCERT. Is it right? The same kind of a diagram is given. You can check the same kind of a diagram whether it is given or whether it is not given. You can check it here very clearly. The same kind of a diagrammatic representation and all the stages which I have mentioned here. What are this? What are this? First one G1, next one S phase, next one G2 phase. What are the things which happens in the G1, S and G2 phase? All these things we are going to study about this. What are the things which are present here? We'll study. First one is called as a G1. What is this G1 and what are the events which happens in the G1 phase we are going to see. G1, cell increases in size. Cell increases in its size. In its size. Why the cell increases in its size? Because if you come to the second point, you find synthesis of majority of cell organelles. Cell organelle. Synthesis of majority of the cell organelles will be done. Next one, synthesis of nucleotides, synthesis of ATP, synthesis of, you find proteins. Everything will occurs in this stage. All this synthesis will be occurring in the which one? G1 phase itself. That's why you find what happens to the size of a cell, because suppose in 100 mitochondria are there, 100 mitochondria becomes 200 mitochondria, then what happens to the size of the cell? Size of the cell has to increase. Endoplasmic reticulum 1 is there, 2 endoplasmic reticulum has become. Golgi complex 1 is there, 2 has become. Chloroplast number has doubled. Everything the cell organelles has doubled. Majority of the minor cell organelles gets doubled in this stage, and also synthesis of nucleotides, synthesis of proteins, synthesis of ATP, everything will be done in this stage itself. It will be done here. This is called as the G1 phase. After this G1 phase, the next stage is called as a S phase. What is meant by this S phase? The S phase which is present is called as synthesis phase. In the synthesis phase, what happens? That we are going to watch it. In synthesis phase, if you come across, we have two important things, because it can be seen in a prokaryotic cell or it can be seen in a eukaryotic cell. Two kinds of cells are there. Prokaryotic cell, eukaryotic cell. But whatever it may be, in the eukaryotic cell, if you come across eukaryotes, we have two important things. In the eukaryotes, we have two important cells. One is a plant cell, another one is called as animal cell. In plant cell what happens, in animal cell what happens? First of all, we will watch in a plant cell. Plant cell what happens? One important thing will be done which is called as a replication of DNA occurs, where 2C becomes 4C. This is the event which occurs in which one? Plant cell. Like if you come for the animal cell, what happens here in the animal cell? Here also replication of the DNA. Replication of DNA occurs. And also apart from this, replication of DNA occurs means 2C amount which is present will becomes in a 4C. See here the C refers to, it's an amount, it is not the chromosome. 2C means amount. I already said in the starting page. Like 20 picograms in the parent cell, now it becomes 40 picograms. 20 will be dividing to one daughter cell, another 20 will be going to another daughter cell. Now you can check it here, the same kind of a thing. Suppose this 2C, you take this one as a 20 picograms, now it becomes how much here? Due to the replication, it becomes 40 picograms, it becomes. has doubled. That is the one event which occurs in the animal. There is another event, the second important thing which occurs here is called as centromere or centrosome. Centrosome duplicates. Centrosome which is present here gets duplicate, means animal cell will be having a special kind of a structures which is called as of centrioles. The centrioles which are present here gets duplicated, means doubled. Doubling of this will be done. That means one cell, you find one will be there. Now in the same one cell, how many it becomes? 2 centrioles it becomes. That is called as a centrosome duplicates. This NEET examination in the last May 7th, you find this question is the one which was asked here. Centrosome gets duplicated in which phase? They asked. It is nothing but which phase? Now you can check it where it is happening. It is happening in the synthesis phase. It's a current. Day before yesterday on 7th, the NEET examination of 2023. This is a question which was asked in the NEET examination. Okay, like centrosome duplicates in which phase? Now you say centrosome duplicates in S phase. And where do you find the centrosome? Only in animal cells, whereas in a plant cell you will not find that one. These are the things which happens. Apart from this, what are the points which are given there that we are going to check it? I'll erase this. Okay, now we will come back here. What are the things which are present here? We are going to watch it here. Check it here. This is the cell cycle. And what are the things which will be present here? What are the things which happens? And G2 also I did not say. G2 I will be giving. You find the dividing further into what phases? Interphase. 3 stages. What are the 3 stages which are present here? One is called as, you find gap 1, synthesis phase. This 2 we have seen. G2 phase, what happens in this? The G2 phase, the things which happens, cell increases in its size. The same kind of a thing. Cell increases in its size. Apart from this, synthesis of proteins, RNA, everything will occur. And also another thing, the special kind of a proteins are synthesized in the G2 phase, which is called as tubulin protein synthesis. What protein synthesis? Tubulin protein synthesis. Tubulin proteins synthesis. Why this tubulin? This tubulin proteins are the one which are mainly useful for the formation of spindle fibers. It is mainly used for the formation of what? Spindle fibers is very important one is this. Spindle fibers will be done here. This is the G2 phase or the gap 2 phase. The things which happens. Now we will come for the overall part, or what are the things which will be done. Some cells do not divide further. Like these are the events which happens when the cell enters, and this entire part which is present here is called as of what phase? The entire part which is present is called as the interphase. This interphase will be taking, and how much time duration? It is of 95% of the total duration of a time interval which is required here for the cell division. Okay, this is the complete interphase. The interphase will be happen. Suppose if the cell has entered into the division, and if it is not having an interest to undergo the division, it exits the G1. This is G1. It exits G1 and go to G0 phase. What do you call? G0, which is also called as quiescent stage, where the cell will not intended to undergo any kind of a cell division. Suppose if it is once again intended to undergo the cell division, once again it come back into the G1 phase. It has to come to S phase. It has to come to again the G2 phase. All these things or the events has to occurs once again. That is, it exits the G0, which is called as the resting phase, but it is not the resting, but it is a metabolically active stage where the cells may undergo sometimes the differentiation. Generally, for temporary time, or it may lost the function of a division, like so many things will be there that we are going to see here. Some cells do not divide further, exit G1, phase to enter into an inactive stage called as quiescent stage, which is called as a G0 phase, which is of the cell cycle. Cells in this stage remains metabolically active. I already said they will be a metabolically active but no longer proliferates. What is meant by the proliferates? They will not multiply. They will not multiply. They will not divide. That is called as a proliferates. Unless called to do so. Unless they are asked like, come on, you undergo the division, then it starts dividing. Otherwise, it will not divide, it will be keeping quiet. Quiet in the sense, resting phase, but metabolically the cell is active. Cell is not dead, it is a functional, it do its functions. It undergo differentiation and it do its functions. That is what? Called to do so, depending on the requirement of the organism. Suppose an organism requires the cells, then you find what happens, it has to come to the division and it has to divide and to produce a more number of cells. That is, example, heart cells are the one of the important thing which are present there. Right. Next one, interval between the mitosis and initiation of DNA replication. The interval between mitosis and initiation of replication. Cells metabolically active and continuously grow but does not replicate its DNA. Continuously the growth will be there, but it will not undergo replication, which is called as of G0 phase or the quiescent stage. Next one, this phase marks the phase of a DNA replication and chromosomes duplicate. Where the chromosomes duplicate here? What is the stage where the duplication? In the S phase, I already said here. This is the phase or the marked phase of the DNA replication and the chromosomes duplication will be done. Amount of the DNA per cell doubles, but there is no increasing in the initial chromosomal number. Whatever the chromosomal number is present in a parental cell, the same kind of a chromosomal number will be present in after the replication also. Why I will be telling one important thing? Just notice one of the important the diagrammatic representation I will be showing. Just watch that one which is present there. See here, this is a cell I will be giving for you. This is a cell. Inside the nucleus is present, inside the chromosomes are there. 1, 2, 3, 4 chromosomes are there. And it is having a what? This is a chromatin which will be a present. This is a chromatin which is present there. Chromatin means it's a thread. How many are there here? Here, number of chromosomes which are present is 4. Number of chromosomes is different, amount is different. Here, the amount of the DNA, is 20 picograms. Number of chromosomes, how many are there? 4. You can count it here. This is a 1, this is a 2, this is a 3, this is a 4. Now you take out this one chromosome out. This is the chromatin which is present there. In the interface it look like this only. It's a chromatin, means double stranded DNA which is wrapped around with histone proteins. We call this one as a chromatin. Now the chromatin is the one which is present here. It's a dividing and the chromatin is present. Now what happens to this chromatin? It undergoes replication. That's what we have studied in the S phase. S phase, what happens? The replication of the DNA will occur. What is meant by the replication? Here this double stranded DNA, another copy of this will be produced here. Another copy. See, previously one thread is present, now 2 threads are there and it is attached to in a common point. Attached to common point. The 2 threads are present. These 2 threads which are present for the common point, that's why the amount means a weight has been doubled. 1 thread now it has become 2 threads. But not 2 single threads as a separate. It's attached to common. That's why here you can check it and another point you can check it here. I am giving an extra. This is the 1, this is the 1 extra, this is the 1 extra, this is the 1, this is the 1 extras which are present here. All what happens here? Previously the blue color is there, now another red color is formed there. That's why the amount has become double. The number of chromosomes which are present will be remained same. The number of chromosomes which are present here becomes the same, but the amount of the DNA gets doubled. That is the point which is present, what we have seen previously. See here, what is the point here? See here, the amount of the DNA per cell doubles, but there is no increasing in the initial chromosomal number. Number of chromosomes 4 are there, the 4 will be there, but the amount of the DNA gets doubled. That is the event which happens in which one? Is phase. Next one, in animal cells, centrioles duplicates. I have given another word which is called as a centrosome. Centrosome contains centrioles. Centrosome doubles, centriole also doubles. That's what in animal cells, centrioles duplicates in cytoplasm. Where does it occur? Where it is occurring is very important market. It occurs in cytoplasm. DNA replication begins in nucleus. DNA replication, where does it happens? It occurs in nucleus. Centriole duplication or centrosome duplication occurs in cytoplasm. These are the two important things. Proteins are synthesized in the preparation for the mitosis while the cell growth continues. This all the points we have studied individually under this one. Next one, this is the complete or the entire part of the interphase. Next to the interphase, we will be going for the another important thing. Next one we come for the next kind of the page which is present. The next one we come for the next kind of a M phase. Now the interphase is the one which is done. After this interphase, what happens? You can check it here also in the diagram which is given here. Single strand is present and the two double stranded are the one which are present here. Here single, how many? 4. The 4 what we have discussed, the same kind of 4 is the one which is given here. You can check the 4 which is given in the diagram, which is called as a early prophase or the late prophase. What is this early prophase, late prophase? That we are going to watch it. See here, M phase. What is the M phase? How much time it requires? Students, how much time is the one which is required? 5% of the total duration of a time which is required here. 5%. Now, what happens? What is this M phase? M phase is called as a mitosis or dividing into the cells. It is the divisional phase, actual divisional phase. That's why we call it's a most dramatic period, drama. You find it will be playing a certain kind of a drama, like all the chromosomes will condensed, they move here and there, they come to one equatorial plate, after that the centromere splits and the movement of the chromosomes to the opposite pole, everything is like a drama which happens here. That's why it is called as most dramatic period of the cell cycle is M phase. Chromosomal number in the parent and the progeny cells are the same, hence it is called as equational division. M phase, which is we are going to discuss the first kind of a stage called as mitosis. Under this mitosis, two important events will be happening. One is called as interphase, another one is called as M phase. M phase is this M phase where the actual cell divides, and here this is considered as the most dramatic period of the cell. Chromosomal number in the parents and the progeny cells is the same, hence called as equational division. How this M phase is divided further into 4 stages, of which the first one I already said here. First one is called as of karyokinesis. Karyokinesis, the first substage which is present is called as a prophase. Pro refers to first. Phase refers to stage. It is called as a what? Stage. Pro means first stage which happens. What are the things which happens in the prophase? First of all, we'll see the first phase follows which one? S and G. S and G2 phases of the interphase. After that G2 phase. After that what will be there? Prophase, marked by the initiation of condensation of a chromosomal material which it becomes untangled. Here you find it is present attached to the coordinates which is called as a tangle. Now it becomes an untangled and it will be condensed to form the chromosome. I already shown in the last picture. This is a centromere and it is having a 2 chromatins will be present because replication of the DNA has happened. Now what happens to this diagram? This diagram is changed to this kind of a diagram. Now this is called as a condensed part. See here. See the difference of this and this. This is just chromatin. Now it is forming chromosome. Condensation of a chromatin to chromosome will happens. That is what which occurs here. This is the first one. Centrosome starts move towards the opposite poles. Centrosome which is present there will starts moving to the opposite pole. Here see, this is a cell, this is a nuclear membrane. Here the two centrioles will be present here. Check it. This is a one centrosome, this is another centrosome. Centrosome got duplicated. That is what I asked for you. And it is the question which is asked in the NEET examination also I told. Two centrosomes are there. The centrosomes are present in cytoplasm. One will be moving into the other direction. One will be present there, one will be moving into the other direction. That's why one is moved and one is present here. It will be moving into the other direction and it initiates the formation of what? It initiates the formation of aster. Aster formation will be done. It initiates the formation of aster. This is called as asters, means microtubule formation will be done. Microtubule formation will be done. It is moved to the opposite poles and it is responsible for the formation of aster. Aster formation. That's what which is given. Centrosomes to move towards the opposite poles. That is the one which is done. The completion of prophase can be marked by what are the things which happens. How to say prophase is completed? How to say? Chromosomes condense to form a compact mitotic chromosome. This is a chromosome. Next one. Each centrosome reaches to the opposite poles and radiates out a microtubule called as aster. I have told centrosomes has moved to the opposite poles and initiates the aster formation. The microtubules will be formed in the form of aster. Aster means star. Like take a globe and take one bulb, which is a bulb. Completely you completely cover within a black paper. Take a ball pen, make a holes for this paper, and then switch on the light. Then what happens? In one direction the light will be passing through. All the holes, the lighting will be coming in all the directions of 360. It's a 3D structure, like in all the directions. Likewise, the centrosome is also in a bulb-like in all the directions inside the cell, because the cell is a three-dimensional structure. Inside it radiates in all the directions. That will be done. Here also it radiates in all the directions will be done. Each centrosome reaches to the poles and radiates out the microtubules called as a aster. The two asters together within a spindle fibers forms a mitotic apparatus. It forms the mitotic apparatus where you find the attachment of a spindle fibers will be done to that. How it will be done, that will be watching. At the end of the prophase, cells do not show. See here, what is the cell organelle? Golgi complex, endoplasmic reticulum, nucleolus which is present, and the nuclear envelope which is present also gets degenerate. Here the nucleus is having a nuclear membrane. You can see it's a fragmented into number of bits. This is initiated. Late prophase means at the end of the prophase, what happens? Complete, you find a breaking of clear-cut breaking can be seen of the nuclear membrane. Clear-cut. And also what are the cell organelle gets disappears? Endoplasmic reticulum, Golgi complex. ER means endoplasmic reticulum. GC, Golgi complex, nucleolus, everything will disappears. Means which cannot be seen at that time. This is called as a latent phase, we can say, where it cannot be seen. See, we know the lunar eclipse and the solar eclipse, or what the moon or the eclipse. When eclipse comes, then we'll be watching like the Rahu, the snake will be coming and it will be eating the moon and everything. It's not eating, like something will come in the middle of that, so that it cannot be visualized to us. Similarly, the cell also, when it is undergoing the late prophase, what happens? The sudden kind of a cell organelle, major cell organelle, it will not be seen inside the cell. It will be latent, means it disappears. See, disappearance of Golgi complex, endoplasmic reticulum, nucleolus, and also nuclear membrane. See here the nuclear membrane is disintegrating. That is what which occurs. And the next point which is present here is metaphase. What is the next metaphase? You can check it here very clearly. See, first one, meta refers to what? Second. Meta refers to second. Phase refers to stage. Second stage which is present is called as metaphase. What is that? The complete disintegration of nuclear envelope marks the starting of the metaphase. How can you indicate that it has entered into the metaphase? Previously we have seen disintegration of the nuclear membrane. Now what happens? A complete disappearance of the nuclear. There is no nuclear membrane inside of this. What is present? Only the cell membrane is present. This is the cell membrane. There is no nuclear membrane inside. Nuclear membrane has disappeared. Completely gone. There is nothing. That's why, what is this? This is a centrosome. This is a centrosome where the centrioles are present. And this is the aster which is radiated. This is the aster which is radiated. This asters will come and attach us to which one? Kinetochore plate of the centromere. Centromere is present for the chromosome. How many chromosomes are there here? 4 are there. You see. 1, 2, 3, 4. 4. But each is having a 2 chromatids. The 2 chromatids are called as a sister chromatids. I will show in the diagram. You check it here. This is the centromere. This is 1, 2 chromatids are present. 2 chromatids. And here there is a one plate. Here there is a one plate. This plate is called as kinetochore. Kinetochore. And this is called as centromere. And these 2 are called as sister chromatids. Sister chromatids. Why they are called as a sister chromatids? Because they are attached to in a common centromere. That's why they are called as a sister chromatids. And what is this kinetochore? The kinetochore is a proteinaceous plate which is present on the either side of the centromere, to which the mitotic spindles will be attached like this. This is called as a mitotic spindles which will be attached. You can see here attachment of a mitotic spindles from the opposite poles. They will be attached. See, inside the nuclear membrane got disappeared. Then what happens to the chromosomes? Chromosomes will be evenly distributed inside the cytoplasm. Hence, in the room, one balloon is present. The balloon was blasted, and the things or the materials which are present inside the balloon, everything will be distributed inside the room. Here and there, in one corner, that corner, like this it will be distributed. All the things has to brought to one equatorial plate. What is that equatorial plate? How it is present? That we are going to watch it. See here, condensation of chromosome is completed and can be observed very clearly under the microscope, and the morphology of the chromosomes are best studied in this stage which is called as M. Remember, M for M. It's a mnemonic like. During a cell division, morphology of the chromosomes can be best studied in which stage? M phase, which is called as, what is this M? Metaphase. Next one, each chromosome has 2 sister chromatids which are held together by a centromere. See, this is a centromere, this is a chromatids, 2 chromatids which are present here. I already said these 2 are called as a sister chromatids, are held together with a centromere. Each chromosome connected by a what plate? Kinetochore plates. 2 spindle fibers from one pole and its sister chromatids connected by its kinetochore to a spindle fibers from one opposite poles, like it will be attached. It is repeated once again. You find the spindle fibers is attached from the opposite poles. The opposite poles you find the spindle fibers will be attached here. It's a very important. The plan of alignment of the chromosomes at metaphase is referred to as metaphasic plate. What is that? All here and there which is randomly distributed inside the cell, they are attached with the spindle fibers and they are brought to one equatorial plate. This is called as a one equatorial plate, one alignment it is brought together, which is called as metaphasic plate it is brought. Why it is brought to a metaphasic plate? It is placed at the center of a cell so that it can equally divide the things into two daughter cells. How it will be dividing that into two daughter cells, that we are going to watch it in the next stage, which is called as anaphase. Ana refers to up, phase refers to stage. The up stage which is present is called as anaphase. What is this? Centromere of each chromosome splits simultaneously and the chromatid separates. You find what happens here. This is a chromosome which is present there. See here, one chromosome I will be giving. This is a chromosome which is having a two sister chromatids. Now the centromere splits. One centromere will be moving here along with the chromatid. Another centromere will be moving into another pole within another chromatid. Two sister chromatids separates in the anaphase. The two daughter chromatids or the two sister chromatids, now referred to as a daughter chromosomes, begins their migration towards the opposite poles. Now what happens to this? This has been split and it has moved to up, it has moved to up. It has moved to north pole, one has moved to south pole. They have moved to the opposite poles. The centromere of each chromosome remains directed towards the poles and the arms trailing behind of this. Now what happens? Centromere is going like this. It's going like this. What happens to this arm? Arms also will trace the pathway of the centromere. Here the centromere is going. These two arms also will be going. Here the centromere is moving. Two arms also will be moving. This is a thing which happens in the anaphase or the up phase. While they are moving, the shapes of the chromosomes can be best studied. That we will study detail in the later classes. The next one is called as a telophase. What is meant by the telo? Telo refers to end phase. What is this telo? Telo refers to end. Phase refers to stage. The end stage which is present is called as telophase. See here the end phase, what is happening? Check it. All the stages what we have seen in the prophase, opposite to that everything happens in the telophase. Do you remember what are the stages which happens in the prophase? Prophase, the first stage, condensation of a chromatin to chromosome. Here decondensation of a chromosome to chromatin. There nuclear membrane disappears. Here nuclear membrane reappears. Their centrosomes move to opposite poles and initiates aster. Here inhibition of aster formation will be done. Nucleolus, Golgi complex, endoplasmic reticulum, everything reappears. Same kind of a points we are going to see. Final stage of the karyokinesis is telophase. Chromosomes that have reached to their respective poles decondense and lose their individuality. Means the chromosome is present like this. Now what it has become? It has become a chromatin like this. It has become a chromatin like this because it has lost, the chromosome has lost its identity. It has become chromatin. Okay. The next one, nuclear envelope develops around the chromosome cluster at each pole forming a two daughter nuclei. You find what happens here, one nucleus, here one nucleus has developed, nuclear membrane has formed, nuclear membrane has formed, inside the four chromatins will be present here, inside of this how many chromatins will be there? Can you check it? 4 chromatins will be there. The 4 chromatins will be present here inside of this. Here also 4 chromatins will be present inside of this. 4 chromatins will be present inside of this. The 4 are present here. That is what which is present here. Now what is the last point? Nucleolus, Golgi complex, endoplasmic reticulum reforms inside of this cell. Inside of this cytoplasm, once again the reforming will be done. That is called as a telophase. After the telophase, next one we come for the next kind of a phase called as cytokinesis. Karyokinesis is done. Next it is followed by cytokinesis. Cytokinesis occurs in plant cell and animal cell. In plant cell how it will be, in animal cell how it will be, that we are going to watch it. See here, it is division of cytoplasm at the end of each cell division gets completed. In animal cells, it is achieved by the appearance of a furrow in the plasma membrane. Plasma membrane, the furrow appearance will be seen. See here, this is a plasma membrane or cell membrane. The cell membrane which is present here, what happens? It forms the furrow from periphery to the center like this. It forms the furrow from periphery to the center like this. It forms the furrow. The furrow which is present here gets deepens. The furrow which is present here gets deepens, and here the repairing of the thing will be done, and the separation of this single cell previously, now it has become in a two daughter individuals. This is called as cytokinesis. Previously it is one, now it has become two. Separation it has become separated. This is very important one which you have to notice, that is called as the telophase. Next, in plant cell, cytokinesis is achieved by the formation of wall which starts within a center to the cell and grow towards the meat to exist the lateral wall formation. How it will be formed, that I will be showing here once again. See here, this is called as a cell membrane. The black line is called as a cell membrane. I am giving in a red line, and this red line which is present is called as a cell wall. The red line which is present I am giving is called as a cell wall, because the plant cells, the cell wall is present. Here what happens, the cell organelle called as a Golgi complex, from center to the periphery there is a formation of certain kind of a wall, and this is called as phragmoplast. This will see there is a formation of plate, and this plate makes the separation of the single cell into two cells. After that the membrane formation will be done there, and the membrane formation will be done in the upper cell, membrane formation will be done in the lower cell. That's what which is given here. The formation of a new cell wall begins with the formation of a simple precursor which is called as a cell plate. Just now I have shown you, that is called as a cell plate. From center to the periphery which is formed here is called as a central plate, represents the middle lamella between the walls of the adjacent cells. This is the walls of the adjacent cells. When karyokinesis is not followed by cytokinesis, means nuclear division is done and it is not followed by the cytokinesis, then what happens? Single cell contains many nucleus, more than one nucleus. That's why it is called as sinusoidal condition, or it can be called as a syncytial condition. A cell which is having a more than one nucleus, then that cell is called as a multinucleated cell. Multinucleated cell in botany we call as a sinusoidal, whereas in zoology we call that one as syncytium. That is the terminology we will be calling. See here it is given already. When the karyokinesis is not followed by the cytokinesis, as a result of which the multinucleate condition arises, leading into the formation of what? Syncytium. Syncytium. Liquid endosperm in coconut, which is called as a syncytial. Syncytium, actual or exact kind of a term which is used in zoology, whereas a sinusoidal, I'll write down the point like a sinusoidal. Sinusoidal refers to multinucleate and aseptate condition is called as a sinusoidal. This condition will be resulted. This is the cytokinesis. Thereby the end of the cytokinesis and the cell division, the single cell has become two daughter individuals. What are the events which has happened? That is called as a mitosis part, what we have seen. There is another division. The mitosis is called as a equational division. There is another division which is present. It is called as meiosis part. What is meant by the meiosis? How does that meiosis will happen? All these things. Before to that, what are the significances which are given for this, that we are going to watch it, of the mitosis. What are the significances which are present? Mitosis usually results in an identical genetical complements in the daughter cells. Same kind of a genetic material which the parent is having, a same will be present in the daughter individuals also. Next one, growth of multicellular organisms. The multicellular organism I already said in the starting. What is that I said in the starting? Like single cell which is present, one cell will becomes many cells. One zygote which is present will undergo repeated mitotic division and forms the millions of cells in our body. Even in any kind of multicellular organism, after the sexual reproduction, the fusion of male gamete and a female gamete results in the formation of zygote. Zygote which is present there, you find it will be, what happens to that zygote? Zygote will undergo repeated mitotic divisions and forms embryo. That embryo, here results in the formation of organism. Like growth of multicellular organism will be done by what division? By mitosis. Cell repair will be done. Mitotic division is in apical and lateral meristem, results in the continuous growth of the plant throughout their life. Next one, in some lower plants and in some social insects like haploid cells also divide by mitotic division. Social insects, which is nothing but honeybees, by mitotic division, haploid cells. Generally, here the apical and the lateral meristem results in a continuous growth of the plant. Apical meristems will come for the apical meristem. This is a ground, this is a shoot system, and this is a root system. Root tip contains root apical meristem, shoot tip contains shoot apical meristem. Say for example, here 100 cells are there. 100 cells divides. Once they divide, how many cells will be produced? 200 cells. 200 cells, 100 cells will retain there only. 100 cells will be sent back to the proximal portion of the plant body where the plant will be growing. We all think, and everyone will be having a false or the wrong kind of a notation, that shoot system, where does you find from where it will be growing? You will be thinking that from ground it is coming up. No, from the shoot tip it is coming or descending the cells to the lower part. Root system, you will be thinking from the ground the root is going deeper into the layers. You are thinking that, and the growth will be happen from here. It's not growth is happening from here, growth is happening from here, the tip of the root. Tip of the root contains meristem. That meristem cells will divide and add the cells to proximal portion. When it is ascending a cells, what happens? This part will be going deeper into the layers of the soil. That is the what which is given. Thereby, the mitosis is the one which is done, which is called as of equational division. What division? Equational division. What is equational division? Once again, within a simple words I will be giving. Single cell of having a nuclear membrane and everything, and it will be dividing and forms the two cells. These are the two cells which will be formed. These two daughter cells are identical to parents and identical to each other. That's why they are called as a clones. What do you call? They are called as a clones. Clones can be called. This is how the division will be done, and this is a one type of a division. There is another division within a half an hour will be completing, which is called as of a meiosis division. What is that meiosis division? That we are going to watch it. Next one is called as meiosis. What is meant by this meiosis? Meiosis is exactly called as reduction division. Reduction division. A simple kind of a thing for the reduction division I will be giving. After that we will be coming here. Simple kind of a reduction division will be coming, and I will be giving a small brief of that. After that the points will be coming. Single cell, it's having a nuclear material, and this is called as of 2n is equal to 4 chromosomes, and the amount is nothing but DNA is equal to 20 picograms is present. It undergoes the division which is called as of meiosis 1, and it forms the two cells. Two cells. Now what happens? In this cell, what will be the DNA? DNA is equal to 20 picograms. And the number of chromosomes which is present, 2n is equal to 4 should be there. How it will be dividing? It will be dividing in another way, where n is equal to 2 will be present. Here also DNA is equal to 20 picograms, and here number of chromosomes n is equal to 2 will be present. That's why meiosis 1 is called as reduction division. Reduction division. It's called as reduction division. Again, what happens again? Again it will be dividing. See, here will come here. Again this one will be dividing. Sorry. Again this one will be dividing. Again this one will be dividing by meiosis 2. and it forms 2 cells. This also will forms 2 cells. 4 cells will be formed totally by the end of meiosis. Each cell here, 4 cells are there now. Each cell DNA is equal to 10 picograms. Number of chromosomes N is equal to 2 will be present. Same is present in all 4 cells. Same is present in all 4 cells. This is how the complete essence of the meiosis. Now we will see what are the steps, what are the things which are present in the meiosis division. Okay? Chalo, we'll come back. We'll come back with this and what are the points which are present and how do the division will be done. What are the division and how it happens, because we have in a less kind of in a time frame. We'll be discussing very detailedly in the class, like one by one. Actually it is of a huge time taking kind of a thing with a diagrammatic representation. Slowly we need to go, but it's enough just a motivation kind of a class. How it will be that we are going to discuss in a little bit in a faster way. It's not like a same kind of a thing, it will be a present, it will be a very slowly when we have in a class like offline class. Chalo, we'll come for this. Meiosis. Meiosis already I said what is that? Reduction division. What happened? Specialized kind of a cells divides that reduce the chromosomal number by half results in the production of a haploid daughter cells. Already we have seen 2n is equal to 4 in the parent we have seen. Now 4 cells when they are formed, what I have given, have you noticed? N is equal to 2 I have given. 2n is diploid, n is a haploid. Now the 2n has half the result and it is called as a haploid daughter cells will be produced. That is the first point. Next one, meiosis ensures the production of a haploid phase in the life cycle of a sexually reproducing. Generally the meiosis is a division which happens in all the sexually reproducing organisms during the time of formation of gametes. When the gametes are formed, at that time it has to undergo the meiotic division. The 4 gametes, 4 cells are formed now, that are nothing but the 4 gametes. 4 gametes, all 4 gametes, number and the amount is same, but the material is different in each and every cell. It's not like in the mitosis we have seen, one parent becomes 2 daughter cells. The 2 daughter cells are identical, they are called as a clone, but here 4 are not called as a clones. You got understood? That's why the sibling bond for the common couple will be a different, because what kind of a gamete, what kind of a sperm, what kind of an egg will be fused, that depends. It's in a God decides. Like the sperm is the one which decides within one of the ova where it get fertilized and the organism will be developed. And that's what I said, like the siblings born for the common couple, same kind of a couple. Like 1, 2, 3, 4 kids are there, all 4 will be a different. Do you find all the 4 will be a same? It's not, except in one condition, that is called as of monozygotic twins, it will be a different. That is what which is present. Now we come for the next one. Organisms, whereas a fertilization restores the diploid phase, meiosis involves the 2 sequential cycles of a nuclear division, that is meiosis 1, meiosis 2. This is one nuclear division, this is another nuclear division. I have shown you just now, but only in a single cycle of replication of the DNA will occurs. Next one, meiosis 1 initiated after parental chromosomes have replicated. That means the interphase will be done here also. What is that interphase? We have seen 3 stages now. Meiosis, first one called as a G1, S and G2. Here also the same kind will be done. After that it enters into the meiosis 1. After that it enters into the meiosis 2. What is that meiosis 1? What are the things which are present in that meiosis 1, meiosis 2? That we are going to watch it in the next kind of the things. See here the meiosis 1. What is this meiosis 1? What are the things which happens in the meiosis 1? Meiosis 1, which is called as a reduction division. Reduction division, you find it's mainly divided into stages which are present there, of which the first one which is present is called as a prophase 1. This is called as meiosis 1, which is subdivided into 4 stages. One is prophase 1, next one is Metaphase 1, next one is Anaphase 1, next one is Telophase 1. Prophase 1 is again divided into 5 stages, which is called as leptotene stage. Next one is zygotene stage. Next one is diplotene stage. Next one is packetene stage. Next one is diplotene. And next one is diakinases. See here, meiosis 1 is divided into 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1. This prophase is again divided into 5 substages, which are called as leptotene stage, zygotene stage, packetene stage, diplotene, and diakinases. That all these stages are this. See, first one we come for. Typically a longer, more complex when compared to the prophase of mitosis. When compared to the mitosis of a prophase, this one is typically considered as a longer phase. Means much duration of a time is required. Why it is? Because prophase subdivided into 5 stages. That's why it requires the more amount of a time. Right? Chalo. First one, what is that? Leptotene stage. What is a leptotene? See, what are all the things you have studied in the mitosis of prophase? Same thing, but in a 5 different stages that we are going to study here. That's all. Chromosomes becomes gradually visible under the light microscope, and the compaction of the chromosome continues throughout the leptotene stage. Compaction. What is the compaction? This is a thread which is present there. Now what happens? This thread which is present here will be condensed to form the chromosome like this, and it is becoming in a compaction. It's becoming in a compaction. It's a condensation part. The same thing. Simply you can say condensation of chromatin to chromosome occurs. Next one. What is the next stage? Substage? Zygotene stage. What is the zygotene? Homologous chromosomes called synapses. They are called as synapses. Synapse is accompanied by the formation of complex structure called as a synaptonemal complex. Synaptonemal complex will be a form. What is that synaptonemal complex? The complex formed by a pair of synapse to homologous chromosomes is called as bivalent. See, for this all bivalence, to understand this all the things, I need to say another kind of a thing. Like it takes a time, but I have to say. See here, otherwise you will not understand the terminology. This is a cell, and inside you find this is called as of nuclear membrane. This is the cell, nuclear membrane. It's having a chromosomes. How many are there? And this meiosis is a reduction. It occurs 2n is equal to 4 chromosomes. This is a 4 chromosomes which I am giving. This is a 1. This is a 1 pair. And this is a another pair. This is another pair. How many are there? 4 chromosomes. These two are same. These two are same. Why we have taken 2n is equal to 4? Because the gametes contains n. N from the male gamete. See, male gamete, female gamete. N gamete, N gamete. N and N combines together to form what? 2n. N and N combines to form 2n. That means if it is a human, take a human, how many chromosomes are there in a human? 23 in male gametes, 23 in a female gametes. The haploid set of the gametes combines together, N and N becomes 2n, and 23 becomes 46. That means 2n is equal to 46 chromosomes are present in the single cell, that is called as of zygote. Gametes will divide and forms. Now what happens here? 2n is equal to 46. That means n is equal to how much? 23. 23 where it is present? It is present in a gamete, male gamete. It is present in a female gamete. Is it right? Like this, one comes from the male gamete, one comes from the female gamete. That's why these two are, one is paternal, one is maternal chromosome. Here also one is paternal, one is maternal. I call this one as a P1, M1, this is called as a P2, M2. Okay. Now what happens to these chromosomes? These chromosomes which are condensed to form what? Condensed to form chromosomes. See here, I will condense that one. This is one pair, P1, M1. What's a P1, M1? These are called as a homologous chromosomes. There is another pair, na? This is another pair which is present there. This is another pair which is present there. This is called as P2, M2. Now totally what is present? 2n is equal to 4 is present. This is a one pair, this is another one. This two, like if you take my left hand, right hand, where the two sister chromatids are there, these are called as a two sister chromatids, these are called as a two sister chromatids. This sister chromatids of univalent combines together for the formation of bivalent. Like when both are combined like this, and if you are watching from your side, how many are there here? Only one. But actually how many are there? Two are there. Two combines, this is called as a synapse. We call as synapse, which results in the formation of synaptonemal complex. Okay. Here you can check it. This and this is combined. When it is a combined, I cannot show you as a three-dimensional structure where it is a combined. But I can show one thing, like this is a one, this is a one, both combines together to form a bivalent. That is what the point is mentioned there. You can check it here, the point which is mentioned here. What is this point? What is this point? You can check it. Synapse is accompanied by formation of complex structure called as synaptonemal complex. Both combines together. The complex formed by the pairs of synapsed homologous chromosomes is called as a bivalent or tetrad. This is 2, this is 2, combined together 2 plus 2, 4, which is called as a tetrad, which is called as a bivalent. Univalent combines, bivalent. This bivalent, which is also called as a tetrad, means 4 chromatids are present. That will be done. Leptotene and zygotene are relatively short-lived compared to the packaging stage. This is a short-lived, this is short-lived. After this, the next stage which is present is called as a packaging stage. What is meant by the packaging stage? Here, 4 chromatids of each bivalent becomes a distinct and clear appearance as a tetrad. The appearance as a tetrad, you can see the diagram which is given in the next page. Not this. See here, the diagram is given. This is the first one, the prophase one. Prophase one. See here the combining is taking place. You can check it in this diagram. Check it in this diagram. What is happening here? Combining. This two. Combining, when it is condensed to form, what happens? It is completely condensed. The prophase one, the combining forms the synaptonemal complex, the same kind of points which we are studying the previous kind of a thing. See here, the combining is done. Next one, 4 chromatids of each bivalent becomes distinct and clear, appears as a tetrad. They appears as a tetrad. Appearance of recombination nodules and the site at which crossing over occurs between the non-sister chromatids of a homologous pair. You find what happens here. The crossing over will occur. Univalent combines. After combining, what happens? The crossing over occurs between the 2 non-sister chromatids of homologous pair. Homologous pair, you find the non-sister chromatids which are present will be combining here. That is what which occurs. Next, crossing over is exchange of genetic material between the 2 homologous chromosomes, and it is enzyme mediated process. What enzyme is required? Recombinase is an enzyme which is required here. Next one, the crossing over leads to recombination of genetic material which is completed by a pachydian living in a chromosomes at the site of what? Crossing over. Complete, you find the crossing over part will be formation, a recombination nodule will be developed, and here the pachydian stage is the place where the recombination will be happening. Genetic material exchange will happens. When the exchange of the genetic material is done, then what happens? This genetic material is useful for the formation of gametes, and all the 4 gametes which are produced will be different. This is the one which paves the way for the genetic evolution. That's why the siblings born for the common couple will be different from one to another. This is a pachydian stage. Next to the pachydian stage, we come for the next stage which is called as a diploidine stage. In the diploidine stage, what happens? See, this is all the points wise and that stepwise within a diagrammatic representation to be shown. It is a little bit a difficult part. Beginning of diploidine is recognized by the dissolution of a synaptonemal complex and the tendency as recombined homologous chromosomes of the bivalent to separate from each other except at the sites of a crossing over. Recombination is done. Now this synaptonemal complex separates. When it is separating, all the part of the chromosome gets separated, but at the place of crossing over it will be held like this only. And you can see what is this letter or the shape of this, my 2 fingers, which is showing what kind of an alphabet it shows. It is called as an X-shaped kind of a structure. This X-shaped kind of a structure is called as chiasmata. What do you call? Chiasmata. Chiasmata appearance will be seen in which stage? Diploidine stage. That is what which is given here. You can check it. The X-shaped structure called as sites of a crossing over is called as a chiasmata. Chiasmata can be observed in diploidine stage. In oocytes of some vertebrates, a diploidine stage will be lasting for months to years. That's why the stage is called as dictyotine stage. It will be lasting for few months to years. That means it will be present as a recombination happening. It will be present as such, and it will be lasting for months to years. That's why you find it is called as what kind of a stage? Dictyotine stage under the endukante it will be lasting for few months to few years. That's why the prophase 1 is considered as a maximum time consuming and time taking longest period. Okay, this is prophase. Next one, we come for the last substage of prophase 1. What is this? This is marked by the terminalization of the chiasmata. Terminalization of chiasmata, it is marked by. The chromosomes are fully condensed and the meiotic spindle assembled to prepare the homologous chromosomes to separate by the end of the diakinesis. Nucleolus disappears, nuclear envelope breaks. Diakinesis represents the transition to the next stage, which is called as metaphase. Prophase is done. Next it will be going to metaphase 1, anaphase 1, telophase 1. What is this metaphase 1? See here, prophase, metaphase, anaphase 1, telophase 1. These are the next stages. Up to here, prophase 1, where the 5 substages we have seen. After this, the next stage is metaphase. Metaphase, what happens? Nuclear membrane disappears. That's why the chromosomes are moved randomly. What we have studied previously, same kind of a thing. And the attachment of a spindle fibers will be done. And here also the attachment of a spindle fibers, separation of the sister chromatids will be done. Separation of sister chromatids which are moved to the opposite poles will be done. As a result, here the 2 are there, here the 2 are there. That's why this is n is equal to 2. N is equal to 2 will be formed here. You can check it here, the telophase. The nuclear membrane and nucleolus reappears. Cytokinesis follows. This is called as dyad cells. What is this cell? This is a dyad cell. This is a dyad cell which is present there. And before to this, we have some other kind of things which are present there. What are this? Metaphase 1 and anaphase 1. What are the things which happens in the metaphase 1 and anaphase 1? See here, the bivalent chromosomes aligned at the equatorial plate. They are brought to an equatorial plate here. You can check it. This is an equatorial plate. Two equatorial plates. Microtubules from the opposite poles of the spindle attaches to the kinetochore plates of a homologous chromosome. From here, attaching here, attaching from here, attachment here, attachment will be done. After the attaching, this two will be going to north pole. This two will be coming to south pole. Next one is called as anaphase. Homologous chromosome separates while the sister chromatids remain associated with this. See here, for each chromosome you find the two sister chromatids are there. Two sister chromatids will be attached like this only, they will not get separated. But you find the two homologous pairs which are present here. In each pair, one will be going to north pole, one will be coming to south pole. You can check it here. This is a paternal, this is a maternal chromosome. This is a P2, this is M2. P2, M2, in that pair, P will be going to north, M will be going to south. In this P1, M1, P1 will be going to north, M1 will be coming to south. It may be changed sometimes also. Sometimes the P1 can combine with an M2. M1 can combine with a P2. That also may happen. But here the separation will be done and the two are separated. That's why previously here 2n is equal to 4. Now it has become what? N is equal to 2. N is equal to 2 in this cell, N is equal to 2 in this cell. It's a separated. This is homologous chromosome separates while sister chromatids remain associated at their centromere. Next one, the nuclear membrane and nucleolus reappears. Cytokinesis follows and this is called as a dyad cells. These two cells at the end of meiosis 1. Meiosis 1. The end, the two cells which are for the present is called as a dyad cells. The two cells are called as what cells? Dyad cells are the one which are present there. Next one become for another important thing. Okay. What is the next one? Interkinesis. Next to that there is another stage which is called as a interkinesis phase. What is this interkinesis phase? Interkinesis stage. It is the stage between the two mitotic division, meiosis 1, meiosis 2. Another stage which is present is called as interkinesis. Means interphase మాదిరిగే ఇది కూడా ఇంకొకటి మధ్యలో ఉంటదన్నమాట. It is the stage between the two meiotic divisions. It is generally in a short-lived, no DNA replication. It is followed by prophase 2. It is followed by the next or the immediate kind of an event which is called as prophase 2. See here, this is called as the prophase 2 which is present there. See here, the two dyad cells are there which we have taken from the continuation of that. Now what happens to these two dyad cells? Prophase 1, it is initiated immediately after cytokinesis. The nuclear membrane disappears by the end of the prophase 2. Chromosomes becomes compact. Chromosome once again becomes compact. Next one, the metaphase. Metaphase, what happens? Chromosomes aligns at the equatorial plate. Same kind of things. Microtubules from opposite poles of the spindle fibers get attached to the kinetochore of a sister chromatids. They will be attaching. See here, this is the attachment. See here, this is the attachment. This is the attachment. Here also this is the attachment of the spindle fibers on the both the ends. Now here what happens? Splitting of this is called the anaphase 2. Anaphase 2, what happens? Splitting of sister chromatids. Sister chromatids happens in the anaphase. See here, splitting of centromeres allowing the chromosomes to move to the opposite poles of the cells by short of microtubules attached to the kinetochore plate. By shortening of this microtubules, what happens? Splitting of the centromere will be done and the two sister chromatids gets separated here in the anaphase 2. Very, very important one. Normal mitosis division lo anaphase lo splitting jarugutadi. Ikkada meiosis division lo anaphase 2 lo matrame you find the splitting of the centromere jarugutadi. Anaphase 1 lo splitting of the centromere jaragadu. Adi inkoka important point, you have to mark it. Another important thing, Now we come for the next one, which is called as a telophase 2. What is the telophase 2? What are the things which happens in the telophase 2? See here, meiosis ends within a telophase 2. What is that? Two groups of chromosomes once again get enclosed by the nuclear envelope. They will be enclosed by nuclear envelope, which is very, very important. Cytokinesis follows, resulting in the formation of tetrad. What happens here? Cytokinesis will happen here. What is this cytokinesis? See here, the two cells which are present here, splitting of the sister chromatids will be done. Again you find what happens, the four cells are the one which are formed. Four cells lo ప్రతి ఒక్క దాంట్లో ఎంతో ఏముంటాయి చెప్పండి, chromosomes. n is equal to 2, n is equal to 2, n is equal to 2, n is equal to 2. All n is equal to 2. But all the cells which are present are different. We cannot call here once again as clones. Why we should not call once again as clones? Because here the genetic recombination or the crossing over has happened in the packaging stage. That's why all the four cells which are formed here will not be a same. Number of chromosomes might be a same, but the nuclear material which is present inside of that will be a different. All the four gametes, suppose these are all sperms are different. Suppose this is ova, all four ovas which are present are different, not same. All four are different, which is a very, very, very important thing. Is this is the end of the telophase 2. Next one after the telophase, what are the significances of the meiosis? What are the importance or the significances which are present for the meiosis? That we are going to see. Conservation of a specific chromosomal number, each species is achieved across the generation in a sexually reproducing organisms, paradoxically results in the reduction of the chromosomal number by half. Generally you find the male gamete, female gamete comes from the male organism, come from the female organism. Male organism is diploid, female organism is diploid. But the gametes which are produced from the male and female will be haploid and haploid. Haploid combines together for the formation of diploid, where the restoring of the chromosomal number equal to that of a parent is done. Restoring of the chromosomal number equal to the parent is done there. This is how the significance is the one which is present. Increasing a genetic variability in the population of the organism, which is an important kind of a process of evolution. For every a multicellular organism which is undergoing a sexual reproduction, where what will be happen? Organism which is present there will show a genetic recombination. As a result, it's in a process of what? Evolution. Evolution means a new organism which is not similar to other one is produced. You see the population, how much population now? India is the first in the population throughout the world. We have crossed even China. Even though 142 crores of the population, even though it is present, like one is different from other. This is because of the meiosis. This is because of meiosis. Meiosis also, crossing over is the one which is resulted. Okay. Thank you very much. Thereby the end of the cell cycle and cell division. I might be going in a little bit fast, but there is no time for us. Like in the detail to be discussed, it will be taking a much time. It will be taking 2 to 3 days of each division. Okay. But here overall a brief account of that one I have given. Thank you one and all. Thanks a lot for listening to my lecture. Thank you very much.